We're excited to welcome you to the Technology Symposium featuring GeoMx Digital Spatial Profiling and nCounter CAR-T Characterization, focused on new approaches for CAR-T cell therapies. My name is Sarah Church, and I'm a senior scientist at NanoString Technologies. We have some exciting presentations today from collaborators at the University of Pennsylvania focused on investigating CAR-T in cellular therapies and hematological and solid malignancies. NanoString Technologies has been developing tools to help researchers address some of the challenges in the CAR-T and cellular therapy field. We've been collaborating with key opinion leaders in the field to establish 3 areas of challenges for CAR-T biology, including CAR-T discovery and development, manufacturing, and post-infusion. For CAR-T discovery and development, we focused on bulk gene expression panels that cover characterization of T cells and cellular therapy targets, including immune status and cell types. In addition, we have panels that examine how cellular therapies may be modulating the immune contexture of the tumor microenvironment. Additionally, we have spatial techniques with our GeoMx Digital Spatial Profiler that allow us to look at humanized preclinical models, particularly looking at CAR-T cell infiltration, as well as different changes in the tumor microenvironment spatially. For the manufacturing portion, we have focused on robust understanding of CAR-T mechanisms and biology at the molecular level of each step of the manufacturing process. As I'm sure you all know, there are many quality steps for the manufacturing CAR-T process that provide some challenges. In the apheresis phase, the challenges include the patient samples coming in are highly complex, and viability and purity of samples are of utmost importance. At the selection and activation phase, the challenge is to assess activation status and decide whether further development of CAR-T samples is worthwhile for that particular patient. At the gene transfer step, addressing the efficacy of the gene transfer and effective gene introduction are also very important for the patient's product. Lastly, in the cell expansion phase, examining the fitness of and the expansion of the CAR-T cells, as well as the sustained response of those, are highly important. So to really address these manufacturing challenges, NanoString developed the CAR-T Characterization Panel, which contains content pertinent to each of those manufacturing steps. In addition, there's an option to add in a custom spike-in of up to 55 genes that can include your specific transgene insert for your CAR-T cell. So the last challenge that we really tried to focus on was the post-infusion characterization. Similar here to the discovery and development challenges, we have a panel to address response biomarkers, as well as mechanism of action during therapy. Additionally, we have their spatial technology with GeoMx Digital Spatial Profiling, where you're able to spatially examine and characterize tumors post-CAR-T infusion to answer questions such as mechanism of action, changes in the tumor microenvironment after therapy, how well cells traffic to the tumor, and potentially identify biomarkers of response. So the next phase for our product development at NanoString is really to expand the scope of our products to further help CAR-T cell biologists answer the questions that they need to have answered. We have 4 new products that are either have just recently been launched or are about to be launched that really are going to be very integral for further investigation for CAR-T. The first is our GeoMx Cancer Transcriptome Atlas, which uses NGS readout to measure 1,834 genes with targeted content covering tumor biology, immune response, microenvironment, and a broad array of other targets, including cytokines, chemokines, and transcription factors. This product allows spatial examination of immuno-oncology targets in both solid and hematological malignancies. Further expanding on that, our next product is the GeoMx Whole Transcriptome Atlas. This is a complete gene coverage of for both mouse and human that has comprehensive content covering gene-specific probes designed for every protein. Which this is really a great discovery. There's no limitations in just the cancer content. It's really to look at all genes in the transcriptome. The next product that it also just launched is our protein profiling for GeoMx that is new with NGS readout. So this now expands the ability to be able to measure more targets, so over 100 targets simultaneously. And it contains biological content that's flexible with the ability to select different 10-plex modules covering immunology, immuno-oncology, and neuroscience. So depending on your tumor type of choice, you can mix and match those as you would wish. And then our last one is really one that's on our nCounter platform and very pertinent to the CAR-T field, which is our nCounter T cell exhaustion panel. This is a new panel with 785 genes across 47 different pathways involved with immune exhaustion. And that includes immune activation, suppression, immune status, checkpoints, epigenetics, metabolism, and microenvironment. It's really a great way to measure the exhaustion status during CAR-T therapy. So for all of these products, you can find more information on them on the NanoString website. So I'm very excited to get to introduce our speakers today. Both of them are from the University of Pennsylvania, where we've had a very nice collaboration for the past year or so. Our first speaker will be Dr. Ryan Golden, and he'll be speaking about applying digital spatial profiling to capture CAR-T activity in human tissues. Our second speaker will be Dr. Marco Ruella, who will speak about novel platforms for understanding resistance to CAR-T immunotherapy in lymphoma. I know we're going to have a couple of great presentations, so I will pass this on to Dr. Golden.
So welcome. My name is Ryan Golden. I'm a third-year clinical pathology resident at the Hospital of the University of Pennsylvania. I also conduct postdoctoral research studies in Dr. Carl June's lab. And today, my goal is really to tell you some exciting work that we have going on in partnership with NanoString to apply digital spatial profiling to capture CAR-T cell activity in human tissues. So for the disclosures for today's meeting, I have none. And to begin talking about sort of the outline of what we're going to cover and to give you a mind map of where we're going, I have that here. So we want to first review the successes of CAR-T cell therapy, specifically what we've been able to do at UPenn, and then bring about this question of sort of how do we translate that success in leukemia and lymphoma into solid tumors, metastatic solid tumors that really burden the society. And then we want to talk about the central features of the NanoString GeoMx and how we think it could be very helpful to us to accomplish that goal. And then lastly, we want to share some of our data from the ongoing partnership between NanoString and our group. So to begin, sort of like what this starts with and really what this ends with is essentially the patient. Because in chimeric antigen receptor or T cell therapy, CAR-T cell therapy, the therapy begins with the patient and it ends with the patient. So we begin by isolating T cells from patients and then genetically engineering these T cells with a molecule, with a chimeric antigen receptor that couples recognition of a cancer-enriched or cancer-specific antigen directly to the activation and killing response of T cells, of the patient's own T cells. And so we're able to engineer these cells outside the body, expand them outside the body, and then freeze them down or send them back for infusion into the patient. And so this has been a very exciting technology. The technology has led to FDA approval, particularly for the disease of relapsed refractory acute lymphoblastic leukemia. So this disease would have had a very poor prognosis prior to this therapy, but with introduction of this, about 90%, at least in our earlier studies, about 90% of patients were able to achieve a complete remission, with many of them staying there. So this was a significant breakthrough, not just for the efficacy, which is impressive, but also for the way this technology works, and it distinguishes itself from other types of small molecules or immunotherapy. So with these early successes in chimeric antigen receptor T cells, we're very excited in asking how can we take this success and translate this to other types of metastatic disease. And so if you think about the burden of metastatic disease or cancer in the United States, it's substantial. And the burden of cancer that really falls within the spectrum of what has a successful CAR-T cell therapy right now is very little. So we would love to expand this success to other types of cancer, and specifically solid tumors. And this is something our lab is aggressively pursuing right now. So how do you begin to go about this? How do you take this success in acute lymphoblastic leukemia and then bring that to a solid tumor? So one of the initial things you really have to spend a lot of time thinking about is what molecule you want that chimeric antigen receptor to recognize. And ideally, you want something that would, in a perfect world, be cancer specific, and in a real world, likely cancer enriched. And mesothelin is one of those such molecules. And so this was identified first in the mid-90s by a group at the NIH. It's a GPI anchored membrane protein that can actually be shed from cells. And it's normally expressed across a range of mesothelial cells. And though its biological function remains somewhat unclear, it is thought to have a role in cell adhesion, and on the pathology side, perhaps tumor invasion or metastasis. And so why we're interested in this in cancer biology is that elevated levels of soluble mesothelin are seen in a large fraction of patients with either mesothelioma or ovarian cancer. And then furthermore, a number of these solid tumors actually express high levels of mesothelin, which I just have a small immunohistochemistry figure to demonstrate that. So, we're checking a lot of the boxes for what could be a good target for a chimeric antigen receptor T cell therapy in solid tumor. And then lastly, it's been demonstrated that patients who have been vaccinated with pancreatic cancer cells produce an anti-mesothelin response with CD8 positive T cells, indicating that the body is able to mount a response against this cancer-enriched antigen on its with vaccination. So sort of the next step in thinking about this would be to say, well, could we hotwire this and create a CAR that would bridge T cells and T cell killing directly to that recognition of mesothelin on these tumor cells? And so this is something we've been aggressively pursuing at Penn. We have a couple of different major CAR constructs to test this. So one is the SS1 construct, the other is the M5. In general, they have an SCFE that recognizes mesothelin on the surface of the T cell, and then they provide the 4-1BB costimulatory domain and the CD3 zeta chain to provide that classic first and second signal that can drive the T cell response. So you might ask, okay, well, how are they different? You have two of them, what's the difference? And it's really three major things. So one is the affinity. SS1 has a lot higher affinity for its portion of mesothelin than the M5. Secondly, the SS1 is actually a murine SCFE as opposed to the M5, which is contains a fully human SCFE. And then thirdly, they recognize different portions of mesothelin. And this could be consequential in the tumor microenvironment because the way that the mesothelin looks or is decorated by post-translational modifications may vary. And so the performance of these CARs may vary based on those features. And so that's something that, time will tell. And so, these are the two systems we have going, and we have a number of clinical trials ongoing at Penn that test these different CARs in clinical trials. So just to go through a few of the ones we have going and some of the high points, one of the initial studies actually used an RNA, delivered an RNA to autologous patient T cells to express the SS1 CAR. And this was performed in a range of patients with a range of diseases from mesothelioma, ovarian, and pancreatic cancer. And then secondly, once safety was established through that method, a more stable expression method of using a lentivirus to deliver the CAR was performed. And so this was another trial, again, testing the SS1 CAR, but this time using lentivirus. And so with the M5 CAR, we also have trials in this space. So one trial is sort of an aggregate from multiple solid tumor types, inclusive of mesothelioma, ovarian, and lung cancer. And we actually did observe dose-limiting toxicity in some patients from cohort 3, indicating that perhaps the therapy is a little too active in some cases. And so this is something that obviously we're aggressively pursuing, understanding how this happened. And then lastly, we have another trial specific to pancreatic cancer where we're testing the M5 CAR, not just the M5 CAR T cells, not just by IV infusion into the patient, but also by regional introduction of the CAR T cells. So across all of these trials, some of which are completed, some of which are ongoing, there are a number of human tissue specimens that have been collected. And we have tissue specimens from metastatic nodules before infusion of the CARs and after infusion of the CAR T cells. And this is very exciting because then you can ask questions about how this therapy is affecting the microenvironment of the tumor, interacting with it, and how the tumor interacts with the CAR T cells. So I think this brings us to this central issue of like, what can we do and how can we use these tissues for maximum benefit to deeply study the molecular architecture of this tumor microenvironment. And so I have just an image that we've generated in-house using a pancreatic cancer xenograft line. And you can see the CAR T cells in red interacting closely with the pan-CK positive tumor cells. And so you have to ask a lot of questions about how is it that the T cells are interacting with the tumor, and how is the tumor interacting with the T cells, and what features sort of correlate with effective T cell response or diminished T cell response. And so, there's a lot of open ground here. And what we would like is some sort of instrument that could help us address many of these pressing questions. And so if you were to just sit back and think about what would be the criteria for this type of instrument that could help you ask these questions about what molecules might be driving or inhibiting T cell response, you could come up with a checklist. And that's what I have here, just a simple instrument wish list of things we would want. So I think one of the most important ones is something that is effective for formalin-fixed, paraffin-embedded tissues. These tissues are very commonly obtained in clinical practice, whereas something that is frozen or immediately sent to the research lab, it's a little bit more challenging to navigate. And the stability of FFPE tissue is significant. I mean, you can go back to blocks quite a while after they've been produced to then reanalyze the tissue. We would also want something that lets us detect dozens or perhaps hundreds of RNA transcripts or proteins. And so going beyond just that simple IHC for a single protein or a simple immunofluorescence or RNA scope detecting a single gene by RNA expression. And so lastly, we also want something that lets us really finely dissect the gene expression across small regions of the tissue. So it may not be single cell level, but something where we can get an aggregate signal coming from a few dozen cells, say a few dozen CAR T cells that are in a tertiary lymphoid structure, versus a few dozen CAR T cells that are in the tumor versus in the stroma. And so we really want that fine level of analytical ability. And so I think what's exciting is that NanoString has developed a tool that really helps us check all of these boxes. So the NanoString has this instrument called the GeoMx, and it can perform what they've termed digital spatial profiling to look at these dozens of RNAs or perhaps hundreds of RNAs in small regions of formalin-fixed paraffin-embedded tissue. So I have just a brief overview here of how the technology works. And so conceptually, what you're doing is this instrument allows you to stain for a small number of markers on a slice of formalin-fixed paraffin-embedded tissue. So you stain for, say, up to 4 markers, something like a chemical dye for DNA, or an antibody for CD3, or an antibody for CD45, an antibody for pan-CK to mark your tumor. And then the instrument captures an image of this fluorescing tissue. And then you can look and say, okay, well, I found an interesting region here where I have CAR-T cells infiltrating into this tertiary lymphoid structure, an interesting region here where the CAR-T cells are seem to be attacking the tumor. And so the instrument has software that lets you then draw circles and select what regions you're most interested in. So this is where that level of precision really comes in. And so after you've marked the tissue by these morphology markers, you then follow that with either a protein panel that can detect up to or a little over 24 proteins by antibodies, or RNA panels that can detect as few as about 80 genes or as many as 1,800 genes through their cancer transcriptome atlas. So after you stain with the morphology markers, it's sort of a fork in the road, and either or, you do the protein analysis or you do the RNA. And then you move to select these regions. You pick the regions of that tissue that are most exciting to you and would be most reasonable for comparative studies. And then the instrument would then progress. After you've selected these regions, the instrument progresses to illuminate using small micromirrors, illuminating the regions of tissue that you've selected with a small UV light and breaking loose those barcodes from either the antibodies or the RNA probes. And at that point, a small micropipette can remove those barcodes in a liquid suspension, remove those barcodes from the tissue and deposit that in a 96-well plate. So for me, when I first heard of this, I was really in disbelief that you could do this many sort of complicated steps, and it adds up to an effective way of doing gene expression analysis. But I think the validation data really speaks for itself, and it's an impressive tool to help us and then help other groups interested in tumor microenvironment studies. So going back to the central goal, it's really to identify these molecular factors that are either limiting or promoting the CAR-T cell response in the solid tumor microenvironment. And we have effective xenograft systems for studying these questions in the lab. So we have either ovarian cancer or pancreatic cancer xenograft systems that then we can treat animals with mesothelin targeting CAR-T cells to obtain attack on those tumors and infiltration. And then we can perform the types of analysis that the NanoString GeoMx allows you to do and ask questions about how these T cells interact with the microenvironment, sort of what things enable the response or inhibit the response. So these are the systems we have in the lab. And for us to really get them working on the NanoString instrument, we had to solve this key problem, which is that the CAR itself, we don't have a good antibody to recognize the CAR in formalin-fixed paraffin-embedded tissue. So NanoString and ACD and our group, we all partnered together to come up with a way of labeling the RNA with fluorescent markers using RNAscope technology by ACD. And so NanoString really took the ball far with this and spent weeks optimizing a method that would allow them and then subsequently us to label these CAR cells in solid tumor specimens. And so after weeks of optimization, they got a scheme working that looked very promising. It looks promising. And I think you can appreciate here the regions of tissue of this ASPC1 xenograft that are extensively infiltrated by CAR-T cells. And then kind of zooming in a little bit further, you can appreciate the way that this instrument can finally distinguish the infiltrating CAR-T cells from the background tumor. So to perform our first RNA panel testing using the GeoMx, we really needed to select regions of tissue that were quite distinct based on their level of CAR-T cell infiltration. And so we were able to find regions that had very low to intermediate to high levels of CAR-T cells infiltrating into the tumor. And so what this sets you up for is a nice experiment where you can make predictions about the kinds of RNA transcripts that you would see enriched in each of those regions. So for example, in the region in the middle here, in these ROIs that have a lot of CAR-T cells, we would expect abundant CD3 signal, an abundant CAR signal from the WPRE element, which is detected using their technology. And so when we actually perform this analysis using the GeoMx, and performed unsupervised clustering, you can appreciate how the groups with high CAR T cell burden really cluster together compared to regions of really tumor only or very, very little CAR T cell infiltration. And beyond that, the positive controls that we expect to see enriched in CAR T cell dense regions do show very strong enrichment. So for CD3 and WPRE, the element that's in the CAR, both of those are very highly enriched in regions that have the most abundant CAR T cell infiltration. And so I think the, these sort of like sanity checks really gave us a lot of confidence in that the instrument would perform as we expected it to. And then everything else, all of the other transcripts and gene expression information that you pull from that is sort of the icing on the cake of what you can then actually dig into for discovery and for generating new hypotheses to test. So NanoString is, again, they really took the ball far and getting that labeling method working. They transferred that method to us. And so we have that now working in our hands in our lab where we can label DNA and pan-CK and CAR and CD3 all on the same slice of tissue, and then use their instrument to do profiling for those 80 genes that are in their human immuno-oncology panel. And again, as this is successful, we look forward to expanding to their cancer transcriptome atlas. So to kind of briefly summarize where we are and what we're excited about, so we do have these multiple CAR T cell clinical trials in solid tumors that are progressing at our university. And we have this innovative way of labeling the CARs in these solid tissue specimens, not just from patients, but also xenografts. So there's quite a number of questions that we are excited about asking with this technology working. And so some of the spatial features that, kind of the low-hanging fruit that comes to mind would be looking at regions of high versus low CAR T cell infiltration, looking at the impact of tertiary lymphoid structures, and then looking at gradients of molecules that we think might be involved in either promoting or inhibiting CAR T cell infiltration. So we can take serial sections, and whereas we stain for those initial 4 morphology markers on a serial section, we could stain for initial things and use that tissue to kind of guide our GeoMx, our region of interest selection. And then lastly, we're planning to expand to the cancer transcriptome atlas after we see that early, after we gain that early confidence in their human immuno-oncology panel. And so with that, thank you to all of the groups that participate in these projects. So thank you to the patients and families. And so we, kind of for a lot the data that you're seeing today, a lot of the main players here are from our lab, getting mentorship. I get mentorship from Dr. June and Dr. Regina Young, who's one of the scientific directors in the lab. And then the TCSL, or Translational Correlative Studies Lab, led by Simon Lacey. And then I work closely with Betsy, who's one of the scientists who works there. And then she and I do a lot of the staining and the instrument operation together. And then thank you to our NanoString collaborators who are really helping push this project forward, and then giving us a lot of support as we've been getting these studies going. So, and then thank you.
Thank you, Dr. Golden, for a wonderful presentation. I'd now like to hand this over to Dr. Ruella for his presentation.
Good morning, everybody. My name is Marco Ruella. I'm an assistant professor of medicine at the University of Pennsylvania and scientific director of the lymphoma program at the Hospital of the University of Pennsylvania. I would like to thank NanoString and SITSI for inviting me to present our initial work on the development of platforms to understand resistance to CAR-T cell immunotherapy in lymphoma. These are my disclosures. I think the most relevant one is that I have received research funding from NanoString, and I have been a consultant for NanoString. So I think you are all aware of the great success of CAR-T cell immunotherapy, and in particular, anti-CD19 CAR-T cell therapy in the setting of leukemia and lymphoma. And these are just 2 examples of patients treated with CAR-19. In this first case to the left, you can see a patient with acute lymphoblastic leukemia who was treated at the Children's Hospital of Philadelphia by Dr. Mode and Dr. Grapp. And as you can see here, we analyzed the presence of T cells and leukemic cells in the bone marrow soon after infusion of CAR-T cell, you see that early on, there is quite a bit of leukemia, about 50% of the cells in the bone marrow, and no CAR-T cells. But by day 23, there is a huge expansion of CAR-T cells that leads to a clearance of the leukemic cells in the bone marrow. So very potent anti-leukemic effect in the bone marrow. And similarly, in the setting of lymphoma, you can see that a patient with pretty extensive disease, you can see all these lymph nodes before treatment, then shows a complete remission at day plus 21. And these are data that were published by Dr. Schuster from Penn. And thanks to these data and other ones that were collected in the registration trials, CAR-19, and in particular, the 4-1BB co-stimulated CAR-19 that was initially developed at Penn and then developed also by Novartis, was approved by the FDA as the first cancer therapy, the first gene therapy for cancer. This was in 2017. You can see this is how the product look now that is commercially available. And then in May 2018 was approved also for lymphoma. Now, many countries, as you certainly know in the world, have CAR-19 approved. There is also another product, actually another 2 products, other are coming up, and hopefully we will see more indications, including follicular lymphoma, CLL, and etc. But if we look at the curves that led to the approval of Kymriah by the FDA, we can certainly see that the event-free survival or progression-free survival is very good for patients with relapsed and refractory disease. As you can see here, these are patients that would have had a very poor prognosis otherwise. Now we can think that about 50% of them are cured, or we hope they are cured because we see a long-term plateau. And similarly for lymphoma, there are more patients that are not responding early on, but the patient that have a complete remission after 6 months or maintaining a remission after 6 months, you can see that they have a high likelihood of maintaining the complete response. But another way of looking at this curve is to see that there is still a vast number of patients that needs other treatment that will eventually fail CAR-19. So certainly there is a need to study the resistant mechanism. And only when we understand the resistant mechanism, we will be able to develop novel effective approaches. This is from a review paper that Guido Ghilardi in my lab recently published, where we were trying to summarize what are the main mechanisms that lead to failure of CAR-T cell immunotherapy, especially in the setting of lymphoma. And there are many ones, many different mechanisms, including pre-infusion barriers that you can see listed here that would reduce our capacity to manufacturing a product or to use a product. But I would say that the 3 main ones can be summarized here in this yellow box. One is the CAR dysfunction. The T cells are not functional, and so our patients would fail. The role of the tumor microenvironment that can be a barrier to the efficacy of our T cells. And then there are more mechanisms that are directly related to the leukemia or lymphoma cells. And today I would like to focus first on the CAR dysfunction, and then I will touch upon the role of the tumor microenvironment. So a recent paper from Penn, from Dr. Freira and colleagues, show that if you look at the CAR-19 products, you can get a sense by looking at the gene expression about what is the functionality of this CAR-T cell. And in particular, they did that in chronic lymphocytic leukemia using RNA sequencing. And as you can see here, patients achieving complete remission have high memory T cells. They have low exhaustion of T cells, so less differentiated T cell subset, less exhaustion. while the group of patients who didn't respond to CAR-T cells, as you can see here, they have higher hypoxia, glycolysis, higher apoptosis, and so on. And to the right here, you have just an example. If you have the signature for the memory differentiation of our T cell, you can see that the complete responders have higher memory differentiations, early memory or memory cells in their CAR-T cells as compared to the non-responder. And this is another example for exhaustion. As expected, patients with lack of response, they have higher exhaustion signature, as you can see there. So we hypothesized that we can capture these signatures and this characteristic of the T cell by using a more targeted panel and using the NanoString technology that is shown here and has been described, where you're able to do to capture the gene expression of a defined list of genes with a very high efficiency, and it's quite straightforward to acquire and to analyze the data. And for this study, we use a panel that was developed recently by NanoString, including 780 genes, including 8 essential categories for CAR biology. You can see one category is signaling pathway related to T cell differentiation and activity, T cell phenotypic and metabolic switches, T cell subset, TCR diversity, immune cell profiling, and other housekeeping genes that allow us to control for the expression of the other genes. What is also important to note is that we were able to customize this panel by also including CAR-19 specific probes that allow us to measure the transcript of our specific CAR. And so the study included 46 patients with lymphoma, both diffuse large B-cell lymphoma and follicular lymphoma, that were treated at Penn with what is now known as Kymriah, so 4-1BB lentivirally transduced CAR-19. And the data that we analyze at this point, and these are still preliminary analysis, is the best overall response, identified as complete response, partial response, or lack of response. We look at the cytokine release syndrome data, presence, absence, or data not available, and the CAR-T cell persistence as defined as more than 90 days or less than 90 days. And as you can see here, we were able to generate a heat map looking at different pathways here to the in the y-axis, and the patients are listed here. So we look at the clustering based on these pathways, and you can clearly see that there are sort of 3 main blocks where patients are grouped. And at the top of this heat map, you can see information, clinical annotation, including the response, where you can see this color means non-response. Then you have this brownish gray that is complete response, and this light blue is partial remission. You have also the cytokine release information with dark red is no CRS, and this purple is yes CRS, presence of CRS. And then as mentioned, we have the peak CAR, which means that basically the persistence of the CAR less or more than 90 days, as you can see here. So if we look at this data, we can see that there is a group of patients that cluster all together here, which is characterized by mostly complete remission. As you see here, there are a couple of non-responders, but the vast majority is complete remission. They do have CRS, and they tend to have a longer CAR persistence. And if we look at the pathways that are enriched here, we can say that there is T cell activation, There are T cell cytotoxicity, chemokine signaling, and so on. All pathways that indicate T cell activation. While pathways such as the one of apoptosis or glycolysis and so on are reduced. But interestingly, what we also noticed is that there were two other clear clusters that were characterized by overall lack of response. And please note that the majority of patients, unfortunately, treated with CAR-19 in lymphoma do not respond. So this is a very relevant subset of patients to study. And what we see here is that we have two interesting clusters where basically these are the non-responder patients that cluster together based on the presence of cytokine release syndrome here or absence of cytokine release syndrome. This is extremely interesting because it tells you that these two groups are quite different and they might have very different reason why they are relapsing. And to give you an example, some of the pathways that we identify is, for example, the Th2 pathway and the high innate immune system pathway that were present in these different clusters. So in particular, the non-responder, the non-responder with CRS, without CRS, sorry, and the non-responder with CRS. And then we can look at the single genes and do a volcanoplot where you can see what are the key genes that are overexpressing responders as compared to non-responder. And again, this is preliminary. We're looking into this, but interesting, you see that the CAR expression does matter. As you can see here, it is highly enriched in responder patients. Another way to use this data is to look at the correlation between gene expression and presence of cytokine release syndrome. And we do have some interesting results if we look at, for example, there are some chemokine receptors here and other factors that have been shown to be in this study to be shown to be related to cytokine release syndrome. Well, for this first part of my talk, I focus on the analysis of the T cells product on the CAR-19 cells and correlate with outcomes. In the second part of the talk, I want to switch gear and look more at the role of the tumor microenvironment and how we can study in situ the lymphoma microenvironment. And to do so, we use a novel technology that has been described today called digital spatial profiling or GeoMx. And what is interesting about this technology is that you can identify region of interest based on staining with 3 or 4 antibodies and then look into the specific region of interest and check the protein or RNA expression using this machine. And so we started this work by analyzing lymphoma patients, either patients that were treated with chemotherapy or patients that were treated with CAR-19. And we look at different markers, CD68, CD20, CD3, and in order to identify macrophages, B cells, T cells, and of course a DNA marker, and we generated region of interest. And then we use these different modules. One is called immune cell profiling core. The other ones are listed here. Basically, these are key targets for immuno-oncology. They go from exhaustion marker, from co-stimulatory markers, and so on. And what the next slide shows is that we can take a biopsy from a patient with lymphoma, and we can identify using this marker, CD3 for T cells, CD20 for B cells, and CD68 for macrophages, different areas of interest, as you can see here. And we can zoom in and select an area with B cells, an area with T cells, an area with macrophages. And then we can look at the overall expression of the target that I mentioned. Of course, I don't have time today to go into details. This is preliminary, but suffice it to say that as you can see expression of several targets. And if we look at responders and non-responders, we can start looking and noticing some interesting difference. For example, the distribution of the T cells, as you can see here. And then we can take one of these patients and zoom in the different either T cells or B cells and macrophages and look at the protein expression. And again, this is still in the early phase of development, but you can clearly see that there are differences in the expression of these factors in the different clusters, and potentially differences in patients that are either responding or not responding to CAR-19 therapy. So in conclusion, I've shown you that there are new platforms like the one that I described, the nCounter CAR-T cell panel and the GeoMx Digital Spatial Profiling that can really help us looking at the CAR immunotherapy from different perspectives, looking at the T cell quality from one side, and also looking at the tumor microenvironment and the T cell function in situ in our patients. So this work is obviously still in the initial stages, and I'd like to thank Yonggu Lee, who is a postdoc in my lab, who has really leading this together with the NanoString team, in particular Sarah Church and Christina Bailey. This is part of a broader collaboration, as you've seen in the previous presentation with Dr. Junet Penn. And for sure, I would like to thank the lymphoma program, in particular, Steve Schuster and Siddhartha Bhattacharya, who is our hemopathologist. The analysis and really would not be possible to run this experiment and to analyze them without the support of Simon Lacey in the correlative studies lab and Betsy Chang, were really being instrumental for this work. And of course, I'd like to thank the patients and their families, all collaborators in the funding agency, and all of you for your attention.